Well, thank you, Warner. This was a very generous introduction, and I'm really happy to be here. I took yesterday a little bit time off to really reflect about the return for investment of CFAR into my lab. It was a very important investment, not only Warner, who recruited me to the Gladstone and helped me launch my career here, but also, Paul, who has been very, helpful along the way and always there for advice. And when I looked sort of just at the raw numbers, I came up that we actually received 8 fellowships and grants between 2006 and 2019. We have a current supportee or mentee in the program, Ursula, and we also not only received support from the Gladstone UCSF CFAR, but also recently got some support from the Duke CFAR, which was very generous in response to a CFAR, an RFA on tissue immunology and supported our new development in gut organoid development. So really very important, very early investments in projects that are high risk and not yet funded, but have led over time to 26 immediate publications that came out of these fellowships. These are just the publications from the fellows themselves that had directly resulted from the support. If I count all the publications where we cited CFAR, especially the flow core, the CFAR flow core, this is a much larger number. And when you talk about the return, I think this investment was well taken because eventually it resulted in 4 R1s and 1 DP1 Director's Award from NIH, which totals about $15 million. So I think my lab is extremely grateful to the support that we have received from CFAR, and it has taken us a long way. Thank you. So the money has been mostly spent on, our main core focus in the lab, which is the mechanisms of HIV transcription. And I just want to give a very brief introduction here, pointing your attention to this triad of the transcriptional activator TAT that binds to the TAR RNA and recruits the transcription elongation factor PTFB, which is just shown here in its core components, CDK9 and cyclin T1. And we have really worked on all of these components in the past, and its activity on the polymerase that eventually is the engine for the transcription and the activity of HIV. I think we're probably best known for our work on post-translational modifications, where we showed that this is not just a simple on and off process, but is actually modified by multiple post-translational dynamic modifications that fine-tune that process, both in TAT as well as in PTFB and even in the polymerase. And I'm just showing you here the post-translational modifications that we and others have described in the past on the TAT protein, which localize mainly in the arginine-rich motif here, which looks very much like a histone, and some people call it histone mimetic because it receives similar modifications like acetylation and methylation like histone do. We have also focused on, host chromatin. This is the TAT-mediated transactivation is certainly not an isolated process. It occurs in the, context of chromatin. The virus is integrated, and we have, worked very much on modifications of this, of this nucleosome NAC1 here, which is an important factor in transcriptional regulation. And of course, another big factor is the role of host transcription factors that bind to the 5' LTR and that, that play a big role in regulating and connecting HIV transcription to the cellular environment. And today I want to focus on this part in our current unpublished work. So we all know that, a main barrier to HIV eradication is really the persistence of the latent provirus transcriptionally silenced in quiescent, mainly memory T cells. And this is really sort of, one of the last challenges that we're still facing here to push HIV to full success story in terms of treatment. We know that one of the strongest activators of this latent provirus is really T-cell activation. When the cell becomes antigenically activated and starts to proliferate and produce a lot of cytokines, and some people, have, proposed that this might be a way to reawaken the virus and lead to a shock and kill strategy. However, we cannot use T-cell activation because it's too strong of a stimulus, and it will eventually lead to, very dangerous side effects in patients. So people have focused on other, avenues, and one of the avenues that we have taken is, focusing on a transcription factor, FOXO1, which is a master regulator of T-cell quiescence, and therefore very interesting because we know that quiescence is an important characteristic of the latent pool. FOXO1 is, regulating in the quiescent cell is actually in a nuclear state, and when it's in a nuclear state, it regulates all these genes that are involved in cell cycle arrest, reactive oxygen, species detoxification, metabolism, and cell death. And when the cell becomes activated, FOXO becomes phosphorylated, eventually also acetylated and degraded in the cytoplasm, and sort of helps, mitigate activation and gene expression during the activation state. Now, we became interested in FOXO1 because of two recent publications. We were associated with Eric Verdin's publication about the mTOR complex, showing that it's an important factor in controlling activity of HIV transcription. And we also recently published a paper on a factor called SIRT1, an epigenetic factor that controls metabolic reprogramming of CD8 memory T cells. And what we found in that paper was that SIRT1 acts upstream of FOXO1 to control in resting CD8 T cells the metabolism of the state. And what it does is it basically prevents the shift to glycolysis, the spontaneous shift to glycolysis in these cells, and suppresses that these cells become cytotoxic under non-stimulated conditions. However, this only occurs in sort of young CD8 T cells. When they become over age or with a chronic infection like HIV, for example, these T cells become repeatedly stimulated and eventually terminally differentiated. And in these cells, this axis is gone post-transcriptionally, and that unleashes basically the spontaneous shift into glycolytic capacity and cytotoxicity in these cells, and makes them per se already much more aggressive. During the course of these studies, we came across an inhibitor of FOXO1 with an impossible name, and I will just call it FOXO1 inhibitor. But the drug in our hands was very non-toxic and acted at very low concentrations and was rather specific. And when we used this to treat sort of the young CD8 positive T cells with these inhibitors, we could shift them into the glycolytic capacity and cytotoxicity profile of these terminally differentiated T cells, showing us that it's a very powerful tool. So what we wanted to do next is to really see whether that FOXO inhibitor could also, have an effect on CD4 positive T cells, specifically those that are latently infected. And sort of to test this, we took two different routes. First, we wanted to look whether FOXO1 had played a role in latency establishment. And to do this, we collaborated with Eric Verdin's lab and used his dual fluorescent virus, which is different from the regular virus, which usually, or the fluorescent reporter virus that we usually use, which contain the GFP in the place of the NEF gene. And so when HIV is active, and the 5' LTR is active and NEF is expressed, GFP is active, and we can basically see whether with GFP gauge the activity of the 5' LTR in cells. However, in the dual fluorescent virus, we have a second fluorophore, MKO2, which is under the control of a cellular promoter independently from the viral promoter, and just tells us whether a cell is infected or not. And this eliminates basically the need to reactivate the cells to determine whether it's latently infected or non-infected at all, because we can focus just on the MKO positive cells and then see whether they're GFP positive or negative to decide whether they're productively or latently infected. And so when we look at this, we usually have sort of a ratio between latent cells and productively infected cells here in red and in gray. When we now treat the cells with an increasing amount of the FOXO inhibitor, we see that we lose the latently the fraction of the latently infected cells, and we increase the fraction of the productively infected cells, really showing that removing the FOXO1 activity in the T cells leads to a decrease in latency and an increase in productive infection. And this can also be shown in a different way, where we just make the ratio between productive and latent infection, and you see that this ratio greatly increases with increasing treatment. Now, to confirm this, we also turn to CRISPR-I knockdown of FOXO1. Here it's shown with 2 different guide RNAs. We also knock down FOXO3 and FOXO4 because they're very close relatives, to see if there's any cross-reactivity of the inhibitor. What we found is that really only the knockdown of FOXO1 leads to a decrease of latent infection and an increase in productive infection here, while knockdown of FOXO3 and 4 really did not have an effect in this. So next we turn to the question whether, if we go to a cell that already has latent virus established in it, would FOXO have still a role in reversing this or maintaining this latency? And this is just the classical JLAT model, where you have just the GFP gene basically and the NEF gene of the integrated virus, and you can see whether GFP comes on or off to see whether the cell is latent or active. And so these JLAT cells are per se GFP negative or very low in GFP because the virus is latent in them. But when you stimulate them, for example, with T cell receptor activation or TNF, you really activate GFP expression. Now, when we did the same thing with the FOXO inhibitor and waited 24 hours, we really didn't see anything, which was very disappointing. In that time, TNF, as our positive control, really reactivated efficiently. However, when we waited longer, 48 or 72 hours, we actually saw that the FOXO inhibitor over time had a very potent latency reactivating function, even more potent than TNF alpha, for example, after 72 hours. So that really indicated to us, yes, FOXO plays a role in maintaining latency in the T cells, and when we block it, we can reverse it. And also, there's a different mechanism in place between TNF alpha, for example, and the FOXO inhibitor here, because there's different kinetics here. And so when we tested whether both would potentially synergize, we found that indeed when we use increasing amounts of TNF alpha, and combine this with increasing amounts of FOXO inhibitor, we really could see that there is a strong synergy between both, indicating that they might potentially work through different mechanisms. And we tested this with many different JLAT cells, some containing TUT, some not containing TUT. This did not play a role. Viability is very little affected by the inhibitor, and again, we see synergy at various degrees in the different cell lines. So that showed that FOXO, independently from the integration side and the cell clone we have used here, played an effect in controlling latency. But we also wanted to confirm this in primary cells, so we did the same experiment with a dual fluorescent virus here that I showed you initially, and primary T cells, infected them, and saw that, again, if we treated with the FOXO inhibitor, the ratio between productive and latent increased, showing that FOXO inhibition really promotes active transcription and antagonizes latent infection. And we also looked in the more established latency model that Warner has really established here, that we infect resting T cells and then treat them with a FOXO inhibitor. We can also, reactivate the virus in these cells in a dose-dependent manner here. And lastly, we also collaborated with Steve Deeks and Satish pili at the blood center to see whether patient cells would be responsive to the inhibitor, and we found that patient cells responded to the FOXO inhibitor alone with a rather modest increase. However, when we combined it with a low dose of prostratin, mimicking a little bit of the T-cell receptor activation, we really could maximally reactivate the virus in these cells. So for us, this was interesting, but we were particularly interested what is the connection between FOXO and the provirus. First, we wanted to see whether FOXO inhibition would just generally activate the cell, which was not the case because CD69 or CD25 was not induced by the treatment, while CD3 and CD28, which is a mimic of the T-cell receptor activation, really activated this efficiently. So we went into RNA-seq analysis in primary T cells and wanted to see what happens, what gets activated if we treat the cells with FOXO. And we found, 160 genes downregulated, 172 genes upregulated, found a lot of sort of individual candidates, like mainly metabolism genes that were strongly modified, and sort of supported our initial hypothesis that this was done because of metabolic changes in the cells. However, when we took a step back and really looked more globally and looked at what are the upstream regulators or pathways that are really activated, we found that these two, regulators here, ATF4 and TRP3, really stood out. And ATF4 is a transcription factor that is connected to ER stress, and TRP4 is actually a negative regulator of ATF4. And we found that TRP4-mediated transcription was, very strongly disfavored when we treat with a FOXO inhibitor, and ATF4 was strongly, promoted when we treated with a FOXO inhibitor, indicating that there might be really a connection with the ER stress here. Now, ATF4 is a very interesting transcription factor. It really is it is generated in response to stress, viral infection, amino acid deprivation, and ER stress via these very particular kinases that connect the production of ATF4 to these stressors. And what happens is that, these stressors usually lead to the phosphorylation of this translation initiation factor that shuts off most of the translation of cellular genes, but it just promotes very selective translation of certain genes, including ATF4. So when we looked in the T cells that were treated with the FOXO inhibitor, we saw that ATF4 was strongly upregulated at the protein level, but not at the transcriptional level, indicating that there was really a connection between FOXO and ATF4 here. And furthermore, when we looked at whether ATF4 would be binding to the promoter, the HIV promoter directly, we found that indeed after treatment with the FOXO inhibitor, we saw that ATF4 by chip analysis was recruited to the promoter, similar to, for example, the polymerase here as a positive control. Now, the master regulator of HIV transcription in terms of transcription factors is really NF-kappa B, and here I'm showing you the REL-A subunit of the NF-kappa B factor, which is in response to the FOXO inhibitor, really not recruited to the promoter, which was very surprising. In contrast, when we treat with TNF-alpha, the classical activator, we really see a very strong recruitment of TNF-alpha, really explaining why these two drugs are really synergistic, because they really act differently in terms of transcriptional downstream effects. Now, another transcription factor that is very critical for the activation of the promoter and also partially binds to the same site like NF-kappa B is NFAT. And while NF-kappa B was not activated, NFAT was very strongly recruited in response to the FOXO inhibitor. And this actually was recently published by another group in PLOS Pathogens. So we focused on ATF4 because we were really intrigued by the connection with the ER potential stressors that I had pointed out here. And we had, we really thought maybe viral infection or even amino acid deprivation might be the connector to the ATF4 activation and FOXO1. So what we did is each of these kinases that connect the stressor to the production of ATF4 has very specific inhibitors that are well characterized. And so we used all of them to see whether we could modify or inhibit the reactivation of HIV transcription in response to the FOXO inhibitor. And very much to our surprise, we found that the PERK inhibitor, the kinase that was connected with the ER stress, really was the only one that showed us that it had an effect and decreased the reactivation of HIV in response to the FOXO inhibitor, while it had no effect on TNF-alpha. Really, the other ones had exactly the opposite effect, what we showed here. However, you will appreciate that this is not a complete inhibition, it's just a 50% inhibition. So we thought we returned to the finding that NFAT was also very strongly activated and used calcineurin as an inhibitor of NFAT activation, and found that indeed calcineurin also inhibited by about 50% the reactivation of HIV. And when we put them both together, we could indeed completely block the reactivation, indicating that both the ATF4 activation and the NFAT activation are probably critical for the downstream effect of the FOXO inhibitor or FOXO1 in HIV transcription. But are these two really sufficient? To do this, we turn to activators of the pathway. Ionomycin is a drug that mobilizes calcium, and that's the critical step for NFAT mobilization. And fenretinib is an ER stress activator that is currently in clinical trials. Both of them at very high doses are very toxic, but when we combine very low doses of them without much impact on viability, we really can very strongly activate HIV from latency in these T cells up to 60-fold here. So collectively, these data currently lead us to the working model that we have, that we connect FOXO1 in resting T cells unexpectedly to the suppression of ER stress in these cells. And when FOXO is active in these quiescent cells, ER stress is suppressed, and PERK and calcium flux is suppressed, and that leads to the fact that these transcription factors, ATF4 and NFAT, are very lowly active in these cells. And HIV transcription is basically suppressed, and latency is big in these cells or promoted. When we now use the inhibitor here and take FOXO activity out of the picture, we unleash these ER stress signals, which leads to the activation of PERK and the activation of calcium flux, and leads to activation and binding of ATF4 and NFAT to the HIV promoter that drives HIV transcription and decreases latency. So this is currently a working model, and there's definitely a lot of steps still that we have to prove. However, we're very interested, especially in this first step here, how FOXO1 really connects to the ER stress. And how we can further exploit this potentially for manipulating the transcriptional state of HIV in latent or activated cells. So I think what we hope to achieve is to see whether either modulator of the FOXO1 transcription factor or modulator of ER stress of calcium could directly modulate the activity of HIV without changing the quiescent state of the cells. So with this, I want to end and thank my lab. I think specifically Albert, who was really the postdoc who drove this project here. He was helped by Irene, a graduate student in the lab, Daniela, a research scientist in the lab, and Karsten, who was a German intern who was here for 3 months but did a lot of Western blots for us. We collaborated with Eric at the Buck Institute, specifically Rosalba, the two Emily's, Tucson, who helped us with the dual fluorescent virus experiments. Satish and Kyle really were instrumental in helping us with the patient data analysis, and Steve and the SCOPE cohort, of course, for providing the samples. This is my lab's funders. Again, CFAR has been playing a big role in this, and I wanted to thank you all for your attention, and Warner and Paul, thank you again for your service.
So, Melanie, I'll ask a quick question. Do you think that this pathway is best exploited as a shocking agent, or do you think you can exploit this pathway to silence the virus?
Well, I think we're trying both. I think we're really interested both in the mechanism and the translational effects here. I think we're playing a lot also with PERK inhibitors, to see whether we can actually, shut down or decrease, latency, or really promote latency, do exactly the opposite than what I showed you now with the FOXO inhibitor, so that we have a block and lock strategy. But we don't know. I think the patient data are not super convincing in terms of FOXO1 being the drug that should be used as a shock and kill agent, but I think with the ER stressors, I think we have some unique opportunities that we have not fully exploited currently.